I'm going to hand over to our moderator today, who is David Brown, who's the Senior Director for the Science and Technical Innovation from Stealth Biotherapeutics. Thank you, David.
Thank you, Jade, and hello, everybody. Thanks for joining the session today, and I'm pleased to moderate this session on novel therapeutic approaches. As we begin, I'd like to just have brief introductions by our panel speakers before we get into their talks. As a reminder, if you please keep your cameras on, and then you can ask questions in the chat feature. Each speaker is going to speak for about 10 minutes, and then after each of the 3 presentations, then we'll have 20 minutes that we can address each of the questions that are asked, either via the chat function or via those who choose to raise your hand. And I'll mediate that discussion. So for introductions, why don't we go around first, beginning with Jay.
Morning, everyone, or afternoon to some of you on the other side of the pond there. My name is Jay Sarkar. I'm actually a fresh grad out of Stanford, and so I'll be talking primarily on a technology that we developed in my thesis project and now spun out into a company in the Bay Area. And my own background is in biophysics, in fact, and usually the way I view this technology and other solutions in the space is from the viewpoint of systems biology. And so that's a little bit of my background and methodology of thinking. And I'm happy to be here. Thank you.
Thanks, Jay. Lorna.
Hi, so I'm Lorna Harries. I'm a professor of molecular genetics at the University of Exeter Medical School in the UK, so it's afternoon for me. And also CSO, founder and co-director of Senisca Limited, which is a spin-out company from the University of Exeter, built on the back of my research over the last 12 years or so. I run an RNA biology group. We're interested in the interplay between alternative splicing and aging phenotypes.
Thank you, Lorna. And Reenie.
Hi, everyone. My name is Reenie McCarthy. I'm the Chief Executive Officer of Stealth Biotherapeutics. We are developing mitochondrial targeted therapeutics to treat both rare genetic diseases as well as diseases of aging. And we are a clinical stage company with a phase 2 program currently ongoing in dry age-related macular degeneration.
All right, thanks very much, Reenie. So now we'll move into the session where each of our speakers is going to give about a 10-minute presentation about their technologies. And Jay, we'll begin with you.
All right, wonderful. So let's just hope no technical difficulties, but can always keep our fingers crossed. So I'm just going to be giving a rather brief overview of the technology, not too much into the business side. And you have my let me just get this up. You have my email here, of course, if you want to follow up, and we can definitely discuss more of the details. But for those of you, and I think a lot of you may not be familiar with the technology, just wanted to take some time in the brief time we have to discuss just the concepts and the evolution of this technology. So yeah, a little background, as I mentioned, this is a startup about 1.5 years old now out of Stanford University. We're based in Mountain View, around the area where Google is, Facebook is, and it really is focused on what we feel is kind of a paradigm shift in the way that people focus on aging and age-related diseases. And it really comes at the core of how those gene networks that encode or manifest as diseases both degrade and how they could be potentially reset. And you'll see another interesting buzzword in the title there. The use of mRNAs is our particular approach, and is something I imagine a lot of you are hearing about or excited about, given the craze around vaccination and the technology developing for other purposes. So that definitely helps us as well. So just to frame where this technology is coming from, as I mentioned, this was a concept that we developed and we thought of at Stanford. And the idea really comes from the sense that if you're going to try to rejuvenate a cell or a tissue, there are a lot of pathways you can try to understand and try to break down. Or you can also look for if there are already natural working solutions in place. And so this is the starting point, the leaping point for us, because we realize that there is, in fact, one critical junction in the lifespan of an organism, of a species, that necessitates the need for rejuvenation, and that is reproduction. The concept there is simple, that you have aged parents and you need to generate a young offspring. In fact, if they inherit the age of their progenitors, then you will have a progressively aging population with each cycle. And so there has to be some kind of active protocol or active process to reset that, and hence this concept of reprogramming that occurs. So in this phase of fertilization and then the subsequent phases to the embryonic stage, there is this dramatic reset of the cellular epigenetics, and I'll talk to that in kind of the next slides here. But largely the information structure that's encoding gene regulation in the cells are by and large put to a very naive, very early state. And this is what allows a cell to become any cell type in the body, as well as being a youthful version of the parental cells, this youthful embryo. Now, the technology we developed is repurposing or recapturing that technology specifically for resetting the aging parameters, rather than resetting all the information in the cell, resetting the kind of noisy dysregulation that occurs with advanced age. And so this is a technology we've coined as ERA, and it's really, as I mentioned, my own background in kind of systems biology, signal processing. I have a stint in electrical engineering too. The concept really comes that you have an information structure that is building up and accumulating a lot of noise, and the concept of reprogramming or resetting it is to kind of pull out that signal or reestablish that signal from that noise background. And as I mentioned, we're going to be just an overview talking about the concepts, not going to show too much data, just maybe a splash screen, but please feel free to look at our recent Nature Communication paper that came out last year. And at your heart's content, go through the data, contact me if you'd like to talk more, and I'd be happy to speak more on that. And yeah, so the concept really comes from the fact that this is moving from the traditional paradigm that cells are just simple elements in a tissue or simple cogs in the overall system, to actually smart computers. And the actual cell has its own kind of brain in the epigenome. And so this is what a biologist would view the epigenome as. It's this set of chemical modifications, not to the code of the DNA, but the overall architecture of the DNA that is triggered or evolved by cell aging, by environmental factors, and that manifests into gene expression changes and also extracellular signaling changes. So both what's happening in the internal cellular environment as well as its interaction with the external environment are being altered based on all of these stressors that we know as aging. And of course, that fundamentally then manifests ultimately as this dysfunctional phenotype. And then again, to give a little teaser at my background, the same kind of concept occurs if you think about a CPU for a computer, and that you have inputs and outputs, just like these environmental or stressor factors. But in a cell, of course, you have this master regulator, the epigenome, which is taking the underlying coding from the DNA and determining what processes to run. So it's not about what it could do. What it could do is encoded in the DNA, but what it should do. And that's ultimately at the highest level regulated by the epigenome. And then, of course, as we know, that translates to processes and gene expression and protein synthesis. So very evocative terminology, because if you think about it, yeah, if you start viewing the cell as a master computer and there's a master regulator for it, then you can say, okay, if it is programmed in to behave one way, maybe it could be reprogrammed, hence the nice little terminology there. So I want to also stress that this is core to aging in that there's a broader theory of aging, this epigenetic noise, where you're not really driving all of these genes to completely fail, but adding more noise in the regulation of cells. So we think of homeostasis as the natural youthful state that cells want to be in, but as it evolves with aging, and I want to stress core disease mechanisms, you start to see noise in these networks. And I'm just going to kind of overwhelm you with that because it does get very complex. So if you think about aging or age-related diseases, there are a lot of common components of them across different indications, across different tissues. So things like immune dysfunction, fibrosis, cell cycle dysregulation, tissue damage, metabolic disorders. All of these are commonalities in things like osteoarthritis and Alzheimer's and diabetes, and COPD. All of these are fundamental components in the evolution of the diseases. And what I want to stress here, and I've just kind of highlighted in red in all of these boxes, is the core epigenetic component in all of these. And this is ultimately where we have reached as we have peered into behind the curtain and how these diseases evolve, that it's not just a single factor or a single gene that you can turn off or turn on. It's a complex regulation, and that kind of low-hanging fruit arena, or that low-hanging fruit window in which we could intervene, is closing. And we need to have more comprehensive solutions that tackle the gamut of all of these different changes. You can see a mixture of DNA methylation, histone methylation, non-coding RNAs, all of these things create this epigenetic landscape then that degrades with aging and disease. And so very simple, again, just to give you the concepts, one can think about this epigenetic landscape that evolves with age and with function. And this, again, very standard simplification. This is a multi-dimensional space, just putting it on a two-dimensional projection here. And the idea is that you would start out, as embryonic cells that are non-functional. This is your ES cells, your early stages of development. And then you would move towards a functional state here, kind of this valley, this stable valley of youth and functionality. And then these cells are differentiated into muscle, into nerve, into blood vessel, and they serve a purpose, and they know their purpose, and that's encoded in the epigenome. As that further evolves with aging, it's kind of like a slow drift. You don't have muscle turning into nerve, or you don't have bone turning into liver cells or anything like that, but you have a degradation in its ability to actually carry out that function. And so I depict that as kind of a lower potential well here. And so with the reprogramming, it's largely about the ability to, well, I'll say traditional interventions, the concept is kind of pushing it a little bit up the hill, gaining a little bit of functionality, but not really transitioning the state, not really bringing it back to a better homeostasis. And then alternative, where kind of the origins of this technology in embryonic reprogramming, or iPSC, if you've heard of that technology, is to send it way back, kind of erasing all information content in that cell, opening up the potential, getting youth, definitely youth, but losing all function altogether. And so technologies that are in that space are kind of limited in their ability to then execute, because those have to be then reinstantiated in terms of their ability to function. So what we're looking for is this Goldilocks, this sweet spot, and that's what we've kind of focused on developing in originally the academia and now the company in ERA, in this controlled, this regulated, this titrated reprogramming, such that we retain the functional identity, but improve its ability to execute on that. And so this is, of course, the holy grail, the concept of rejuvenation, where you're a functional, youthful version of a cell, as opposed to any kind of transformed or differentiated version. And then I want to stress just briefly on the realities of mRNA reprogramming. This is very exciting to us, again, because of all the architecture that's being built out, because of the vaccine and other technologies out there, but also because it gives us that ability to control. As I showed in the last slide, control is fundamental. You're transforming, or you're evolving the cell in a very dramatic way. You have to be able to very nicely control that evolution. And so the mRNAs give us that ability. You may have heard of other ways that people do with viruses or with transgenes. And the issue there is largely that aspect of control and ensuring that you're not going all the way back to this embryonic stage or any transformed stage in between. So with the mRNAs, it's a very similar standpoint that you're bringing in this mRNA cargo. It's encapsulated usually in a lipid-based carrier, and this is something we can talk about in future discussions, but and also an area of active development for us, where we're driving in this mRNA through natural pathways, and then that mRNA is translating into protein, which then goes back into the nucleus and then resets these epigenetic markers. And so this is kind of spelling it out a little bit more. These are what are known in the field as pioneer factors or transcription factors that largely bind to different regions of the genome and then cause manifested changes in the histone tags or the topological domains, et cetera, et cetera. And then, yeah, as you can see, that's a large scale reset in chromatin spaces and the overall potential of that cell, which when controlled can give you an optimal rejuvenation effect. And so simple time course, we can imagine with the reprogramming, it's a very fast with the mRNAs. The mRNAs, as you imagine, are the reason why they were able to make a therapy so quickly or a vaccine so quickly is because the mRNAs are very fast acting in that you go in, you drive in this nucleic acid, and it immediately starts translating into protein. You can control that expression pattern. You can control that evolution in these different components of the mRNA. So these are all elements that we've designed and optimized for our different reprogramming patterns. And then you can see, yes, you can control the expression lifetime of the mRNAs. You can control the repeatability and the overall time course of the mRNA, of the protein expression. And then that translates to the plasticity of the cell, how it's able to evolve in its state, as well as the efficacy of the reprogramming phenotype. So this mRNA really gives us that ability, more than a transgene or a virus, to really titrate and control how that evolution is happening. And ultimately, that's critical, not just for efficacy, but for safety. And so I'll just kind of summarize here in terms of some of our results. And as I mentioned, this is all visible in the paper. Feel free to look that up. And then, of course, we can talk more offline. But what we've established in this work was that, yes, this is a broad principle, a broad approach that we can apply to multiple different cell types. And you can see a variety of different tissue types, different germ layers. In all of these concepts, we were a stickler about really using the optimal model. So always kind of human cells from primary tissue and from aged donors. No surrogates of progeria, no surrogates from in vitro aging, but really the real McCoy in terms of the model systems there. And then we have a variety of data, of course, all pulling from the paper here, but initially started this broad screen in terms of the hallmarks of aging. I think this is a popular categorization that a lot of you are familiar with, where we did a lot of the different hallmark panels in multiple different assays. And you can see kind of the evolution if we look at two different cell types here on the left and right, fibroblasts on the left, the connective skin layer there, as well as endothelial cells or blood vessel cells. You can see this transition, and this is just, accommodating a lot of data and a lot of single cell data where we're showing the distribution shifts in various parameters, autophagy levels, or sorry, this is kind of blocking my screen, so I can't actually read that. The H3K9, so epigenetic markers, evolving as a population from young to aged, and then with the treatment kind of reverting back, you can see in these different cell types and these different parameters. But as I mentioned, we kind of did the full spectrum. And then afterwards, we wanted to move to more omics analysis, a more holistic view of what's happening as these cells are progressing. And then, of course, we did the transcriptomics, and we saw the gene profile kind of shifting in polarity with aging, and then shifting kind of back with the treatment. The fold change expression, the PCA clustering, all kind of telling us that it is a more transformative and global shift that we're inducing with this reprogramming. And then, of course, moving into disease phenotypes, and there are a lot more now that we've looked at as a company, but just the ones we were hinting at in the paper. Osteoarthritis, of course, a big one for anyone over 60, and looking now at functional markers like collagen production or antioxidant production or inflammation parameters, all showing that kind of improvement with multiple patients with the reprogramming. And then finally, also for injury response is another modality you can think of. There's kind of the degeneracy, but also the damage response. And this was kind of our key kind of leading point at the very end of the paper, that this is a broad kind of phenotype, because we can now do these long-term benefits by hitting stem cell populations in particular. All of these were more differentiated cell types, but with a stem cell population and a directed therapy, of course, you have gains not only after a single injury where you see more tissue being generated and faster improvement or more force output, but even on a second injury, that there are retained benefits when you're hitting this reserve population. So just to largely summarize, as I mentioned, ERA is this method to drive really this kind of holistic reset, but a very controlled reset on the epigenome. And we know that correlates with aging phenotypes as well as these rejuvenative markers that we've seen before. I want to stress that it is very clean and controllable in the platform, and translatable in the fact that we are using mRNAs. And if you've heard a lot of the rhetoric about the vaccine and how safe it is, the fact that you're not manipulating the genetics, that you're not leaving a footprint after the initial delivery of the material. And then really stressing the widespread nature of this reprogramming, and the necessity in general, if you're trying to hit an aging phenotype, to reverse a lot of these interconnected networks and drive a lot of aging hallmark shifts. And then furthermore, with the disease parameters and the age tissue functions, this is what we kind of focus in those last few slides. And then, of course, we're continuing studies. We have a lot of different applications. Primarily, our focus is dermatology. It just provides a very nice test bed for a lot of the indications, as I showed, immune aspects, fibrosis. All of these have a very accessible leaping point when we look at it in terms of the context of skin, and also a very accessible tissue to get at and to evaluate and assay. So these are kind of the first setups. And then, of course, as we're developing into different products, there are other components. We're developing the mRNAs, the delivery solutions with lipid nanoparticles and other solutions like that. But altogether, this concept, we feel, is a true kind of paradigm shift in the way that this aging problem is solved. And so thank you for your time. And hopefully I'm still within, I think I lost my timer in a little bit here, but if there are other questions, you have my email up here. So please feel free to contact me offline, but otherwise I look forward to the discussions and Q&A.
All right, Jay, thanks so much for a really interesting talk. As you stop sharing your screen and Lorna tees up her slides, I just want to remind everyone, I already see some questions in chat, so please either raise your hand or keep those questions in chat going, and we'll get to those once we get to the discussion portion. So thanks again, Jay and Lorna, the floor is yours.
Thank you very much, Dave. So firstly, I'd just like to thank longevity leaders for the opportunity to introduce our company to you. It would be 30 seconds, sorry, I'm changing slides. So we are a new spin-out company from the University of Exeter, which has been founded to exploit our discovery of a new hallmark of aging and to use this to develop a new senotherapeutic approach. And I'm really happy to be here today to introduce our concept to you. So as we all know, aging is caused by the failure of a few basic health maintenance mechanisms called the hallmarks of aging, shown here on the right. And I'm not going to expand on these because everybody here is very familiar with these. These are an interlinked and multifaceted set of phenomena which underpin aging and susceptibility to age-related diseases. And the hallmarks of aging are not only drivers of cellular and organismal aging, they are potential points of therapeutic intervention for treating the diseases of aging. And Senisca is one of a growing number of companies, some of which are represented here on this roundtable today, that are developing new therapeutic approaches based on manipulation of these novel points of traction. So at Senisca, we have discovered a new hallmark of aging, and that's dysregulated RNA splicing. So RNA splicing is the process by which the information in your genes is ordered and rearranged so that cells can appropriately respond to their internal and their external environment. Most genes are actually, in fact, capable of producing more than one message according to the needs of the cell. And the ability to do this is absolutely crucial for cellular health and for successful aging. Now, the decision as to which forms of RNA are produced from a gene in response to any given challenge is made by a group of proteins called splicing factors. And these bind to sequence elements around the splice sites and regulate splicing decisions. And we know that restriction of the splicing repertoire reduces cellular adaptability and plasticity. We've just heard from Jay how important that is. This ultimately induces cellular senescence and other aging phenotypes by virtue of compromised ability to deal with cellular stress. So at Senisca, this is built on the last sort of 12 years of academic research that's come out of my lab, but we've discovered that levels of splicing factors actually decline with age. We've shown this happens in multiple cell types, in multiple species, in population studies, that these things are actually predictively linked with aging phenotypes in populations. And we've also shown that splicing factors are dysregulated in the cells of patients with progeroid syndromes of different types. And we've also unraveled the molecular causes of splicing factor dysregulation and discovered why these splicing regulators are turned off with age. And this has identified several points at which we can actually intervene to restore splicing regulation and splicing patterns to more useful levels. And when we do this, we can bring about cellular rejuvenation. As I've said, this observation holds true in multiple cell types. So we've identified the upstream pathways that lead to splicing factor dysregulation. Also, the effector genes at the bottom of those pathways lead this to happen. And we can target these master control genes specifically to restore correct patterns of splicing regulation and correct patterns of splicing indeed. And this allows us to effectively reset those splicing factors, restore splicing patterns, and to rejuvenate aged cells. And this is a platform technology that could be applied to any disease actually that's driven by cellular senescence. But at Senisca, we're going to be applying this first to diseases with accessible target tissues, which are amenable to local delivery, and to diseases with high unmet clinical need. So, because we've identified the underlying biology and the pathways that lie behind this age-related splicing dysregulation, we've been able to identify and to validate 4 genes, which when targeted can rejuvenate senescent cells. And in fact, if we change the directionality of targeting, we can actually induce senescence in young cells. So there may be sort of oncotherapeutic avenues that we can explore down the line. We also have further data from which we've identified other target genes, which we're now in the process of validating, ready for us to design our specific and novel interventions. So we can intervene either upstream in the pathway, which will allow us to affect many different targets simultaneously, or downstream, which allows very precise specificity to minimize off-target effects. So I'd like to show you just a little bit of data to illustrate the utility of our approach. These data are actually derived from the treatment of senescent dermal fibroblasts with an oligonucleotide designed to induce knockdown of two different master control genes. The first graphic that you can see there is a heat map. This shows that our targeted intervention can upregulate splicing factors. And on the heat map here, green is up and red is down. The targets that we've chosen to manipulate are there on the bottom, and the identity of the specific splicing regulators that we've looked at is there on the side. Following treatment, we see a marked upregulation of splicing factor expression and a resulting drop in the senescent cell load in our culture. And when we assay for markers of proliferation, we see that our treated cells have re-entered the cell cycle, and on the microscope fields that you can see there, that the dividing cells are actually labelled in pink. And we're able to uncouple some aspects of the senescence phenotype. So some of our interventions will cause a drop in senescence and also a re-entry to cell cycle, but some of them just bring about reversal without renewed proliferation, which might be useful in some circumstances. And in fact, some of them actually even lead to rebuilding of telomeres. So we could go after all sorts here, but our initial indication is going to be idiopathic pulmonary fibrosis, known as IPF. This is a disease which is known to have a large senescence component. 3 million people worldwide have IPF, and there is currently no effective treatment. In addition, the drugs that Senisca will design for IPF have potential applicability to other fibrotic lung diseases and to chronic obstructive pulmonary disease, which of course is a much bigger market. And importantly, the currently available treatments cannot stop or reverse disease, but only very modestly slow it, and they don't work for everybody. As I said, this is a platform technology which has applications to many diseases which are driven by senescence. The timeline encompasses 2 complementary approaches. So our main modality is oligonucleotide biologics. These are DNA molecules in this case that bind to specific sequences and disrupt the regulation between our target genes and their upstream regulation. And we have 4 existing targets, which we are currently evaluating our novel interventions for the creation of new IP. But to expand our future scope, we're also currently validating a number of other potential intervention points, because any given oligo may not be amenable to all indications, although we do expect there will be some considerable overlap. Following target validation, we're going to be taking a traditional hit to lead to candidate track through ex vivo 3D cultures, co-cultures, and tissue models, and then following that evaluation in vivo. Our second modality is a small molecule approach applied specifically to skin aging, whereby we are currently undertaking an intelligent computational docking-based screen for molecules that affect our novel pathways. And this is a self-contained and targeted piece of work driven by interest from a potential large US-based potential cosmetic corporate partner. And of course, this also gives us early human in vivo proof of concept. So our team consists of myself as CSO, Kirsty Semple as CEO, and Dr. Ben Lee as CTO. And between us, we bring a wide range of skills in the senescence, RNA biology, and commercial space. In addition, we've got some very big hitters on our scientific advisory board, including Professor Steve Wilton. He's the person who designed and guided the very first oligonucleotide therapy for Duchenne muscular dystrophy into the clinic. And Professor Merlin Thomas, who's currently working on oligonucleotide delivery to the lung, specifically in the context of COVID. We have big ambitions. Longer term, we have several lines of potential development. Our initial indications will be amenable to local or topical delivery, but long term, we have plans to target other systemic diseases. These are obviously higher hanging fruit, but we think within reach using our technologies. And all of the indications that we have here on the slide actually do have a senescence component. We also have ambition to develop specific delivery methods to target our interventions, specifically to senescent cells or to specific cell or tissue types. We also plan to explore novel chemistries for our oligonucleotide backbones, which will allow us to manipulate stability and minimize toxicity. And we're of course open to co-development opportunities or to collaborative partnerships. Indeed, as I say, we already have some of these under negotiation. So I'll finish there. Thank you for listening, and I look forward to our discussion.
Thanks very much, Lorna. What an interesting talk. Reenie, you can go ahead and tee up your slides. Thanks everyone for the questions that are coming in through chat. And after Reenie finishes, I look forward to having a great discussion. So Reenie, the floor is yours.
Yeah, thanks, Dave. And really thank you to my other panel participants. I'll try to keep this somewhat brief so we can leave some more time for questions. So I may be skipping some slides in an effort to do that. At Stealth Biotherapeutics, we're focused on mitochondria, the powerhouse of the cells. We're a clinical stage company, we're publicly listed, so draw your attention to our forward-looking statements. So if you think about mitochondria, exist in almost all of our cells, produce 90% of the energy our bodies need to fuel normal, healthy organ function and human life. We're focusing really on the organ systems, which are the highest consumers of mitochondrial adenosine triphosphate, or ATP, which is the cellular unit of currency. So although our brains only take up about 2% of our body weight, they consume 25% of our energy. And in the brain, the visual system is the biggest consumer of ATP. So we are in phase 2 clinical trial in dry age-related macular degeneration, which is the leading cause of progressive blindness in elderly patients in the Western world. My slides seem to be having a mind of their own and advancing on me, so ignore that if I flip you back and forth. Heart failure has a strong mitochondrial underpinning. We are focused on some rare metabolic cardiomyopathies in the clinic now with Barth syndrome and moving into Duchenne. But we do have data, human data in heart failure as well, which again really verifies this is a promising target for mitochondrial therapeutics. And for neurology, we have a program moving into phase 3 with elamiprotide, our lead compound, but we've rationally designed 2 new classes of compounds to target the brain and have some promising preclinical data in different indications. So mitochondrial dysfunction is known to increase essentially with the aging process. So it can be genetic, you can be born with it, but for all of us as we age, our mitochondria start to become dysfunctional. We have spontaneous mutations in our mitochondrial DNA, oxidative stress generated by those dysfunctional mitochondria, or from external factors like pollution or smoking or obesity starts to damage mitochondria. And so they're literally implicated across the spectrum of human disease, and particularly diseases of aging. If we think about mitochondria as a network within our cells, and they do network in our cells and across organ systems, an important thing when you think about a power network is isolating and reversing damage before you get rolling blackouts. And so why does that happen with mitochondria? Well, you look here at the mitochondria in yellow, and you see that they're constantly dividing and fusing with one another. And that's so that they can dynamically produce our energy needs. So connected network, and once you start to continue to explode. I'm showing you here mitochondria undergoing oxidative stress. And so you can see them kind of ball up and die. The inner membrane, those stripes start to go away. And essentially this oxidative stress causes more and more oxidative stress within the body, which is the leading cause of fibrosis and inflammation across multiple disease areas. With our lead compound, elamiprotide, what I'm showing you here is essentially oxidative stress in a cell. And you can see that if you have a protective agent, elamiprotide, on board ahead of that oxidative stress, you can protect against the cell death. So that's an important takeaway, I guess, that I want to give you from my talk today. When in the course of a disease like aging should we be intervening? there may be a tipping point, right, where at some point you can't necessarily reset the clock. This is really a close-up of mitochondria, particularly the cristae architecture, which you can see in 3D. So these are essentially tubules, and that's really important because within the cristae of the inner mitochondrial membrane is the complex machinery that the mitochondria use to produce ATP. The food we eat, the air we breathe is converted essentially into electrons that are passed through this electron transport chain, kind of like an assembly line, and at complex 5, ATP is generated. The curved architecture of the cristae is really important to keep these complexes close together and minimize the leakage of electrons, which results in oxidative stress. Cardiolipin, depicted in orange on these slides, is the target of our lead compound, L-mipritide. It's a phospholipid, which is essential for essentially this normal mitochondrial intermembrane architecture. And what we see with disease with oxidative stress is that cardiolipin and other key components of the inner membrane and the cell become degraded, leading to that loss of inner membrane architecture, a drifting apart of the electron chain complexes, and more oxidative stress. So this is a vicious cycle whereby oxidative stress leads inevitably to more oxidative stress and disease. What we see with L-mipritide, and you're looking at a mouse model of diabetic retinopathy. In the middle, that is a sick mitochondria in this model. And on the right, after 6 weeks of L-mipritide therapy, we can see restoration of normal mitochondrial morphology. I'm going to show you a quick video to really focus on the networking, which is important. Again, the power grid kind of concept. In red, you have disconnected cristae, so they're not well networked. And on right, with L-mipritide in an ischemia reperfusion injury model, we can see restoration of that normal networking function. So I'm going to take a moment to talk about that question of when we intervene in disease. If you know that an injury is going to happen, and it's not often that we know that, although aging may be one context where we do. If you can intervene with a protective agent ahead of the injury, that may give you your best shot at preventing ensuing disease. So this is a good example. This is percutaneous transluminal angioplasty, where you're going in to clear a blockage in the renal artery. In animal models, we know that the reperfusion injury, as the blood rushes back through that now cleared artery, can damage the mitochondria and lead to damage to microvasculature of the kidney. This is data from a phase 2A clinical trial that we conducted at the Mayo Clinic with elderly patients undergoing PTRA. And on the left, you can see that placebo-treated patients post-intervention did have a lot of hypoxia. That's in yellow and red on the images, whereas L-mipritide was able to prevent that, and then that led to better perfusion. Unfortunately, in most cases, when we're treating diseases of aging, or even the aging process itself, we're intervening later in the game after the injury has already occurred. And so really, I'm showing you the example of dry age-related macular degeneration, leading cause of blindness. It's progressive, and this is the leading cause of blindness among elderly patients. Essentially, it leads to areas of cell death in the retina. And once these retinal cells die, they cannot be regenerated. In a phase 1 clinical trial, so no placebo control, in patients with dry age-related macular degeneration and those areas of cell death or geographic atrophy, after 6 months of L-mipritide treatment, we would have expected to see about a 2 to 6 letter decline in vision, just from the natural history. Instead, we saw that patients in both their regular light conditions as well as essentially night vision or dusk conditions improved by about 5 letters in vision. And those improvements were also associated with improved visual quality of life for these elderly patients. So new data that was just presented last weekend at ARVO is really an analysis of how these improvements in vision relate to mitochondrial health of these patients. The ellipsoid zone is an area of the retina that is very mitochondria rich. And what you're seeing in pink or purple is bad mitochondrial health. So this is baseline. At baseline, the patient on the left-hand side of this slide had a lot of attenuation of his ellipsoid zone. So not much mitochondria left, and only a 2-letter gain in vision. And over to the right, we see that a patient that has less attenuation of the ellipsoid zone, particularly in the fovea, which is the center of the eye controlling vision, was able to gain many more letters, 18 letters. So again, these slides are really kind of going to a theme of, is there a tipping point in disease? After which it will be hard to kind of reverse dysfunction. I will just kind of say briefly that for cardiology, there's strong support for mitochondria as a target in cardiology. There's broad consensus on this. We've seen in explanted human heart tissue from heart transplant patients that L-mipritide can improve mitochondrial function. We saw trends toward improvement, particularly under maximal stress in patients in a 1-month trial in heart failure with preserved ejection fraction. And for us, we're focusing on some rare metabolic cardiomyopathies such as Barth and Duchenne cardiomyopathy. The brain is another area where mitochondrial function is a clear therapeutic target. With L-mipritide in preclinical models of Alzheimer's as well as senility, there have been benefits. Our next generation clinical compound, SBT272, has shown improved neuroprotection. This is in a TDP43 pathology, which is implicated in ALS, but we've also seen that in other models. So again, our approach is really that with mitochondria being almost ubiquitous in human health and disease, we've learned a lot of lessons over our years of clinical development with this approach. And we do have clinical data in many age-related diseases. So we do think that this is an interesting approach to the aging process. Big question will be trial design and really regulatory pathways for aging. So with that, I will stop sharing my slides and hopefully open up to questions.
Thanks so much, Reenie. So really interesting talk from all three of our panelists. At this time, we continue to invite everyone to ask questions via the chat feature. I know that we have some questions coming in already, so let's get the dialogue started. One question, let's see, this would be for Lorna. Since senescence is an anti-tumor strategy, can re-entry into the replication cell cycle be potentially tumor promoting?
So you'd think so, wouldn't you? But actually, because of the nature of the regulation of our targets, we find no, that it's a homeostatic mechanism. There's a great big negative feedback loop. So if you upregulate them to a point at which that might be at risk, the feedback loop switches them back off. So it's all kept in a really narrow homeostasis. And in fact, actually, some of our interventions don't lead to re-entry to cell cycle. So we know which bits of that pathway to hit to actually get reversal, but not reinitiation of cell cycle. And in fact, some of the tool compounds we use to actually unravel this in the first place are actually in clinic as anti-cancer drugs.
Excellent. Jay, do you agree? Similar thoughts for you that about the potential tumor effects of reversing some of the senescence?
It's definitely an interesting area for us. I was talking to Lorna a little bit about this, that we, for our reprogramming as well, it's something that we're very careful of because, you know, the technology for us is very transformative too. And we don't want to re-engage any potential, so on our end, you know, the way that we're approaching it, re-engage senescent cells to become potentially cancerous. But strategies like Lorna is describing, where you can maybe address the senescence population first, and then come in with, you know, maybe a broader theme of synergies between technologies, but come in with a reprogramming afterwards would be really interesting if those two could work together in that sense.
Thanks, Jay. Got a question for Reenie. Has L-mipritide been entered or used in NIA drug testing mouse trials?
So there's been a lot of work done at the University of Washington, and those are under NIA grants in aging models, but not specifically in NIA drug testing mouse trials. There's been work done in aging models of heart failure, aging models of ophthalmic disease, and just aging mice generally. And there's just a whole body of work with Peter Verbinovich and Dave Marcinick, again, out of the University of Washington.
Excellent. At this time, are there other questions? We're just about out of time. Are there other questions from the panelists? Anyone like to enter a chat question or raise your hand at this time? Okay, so I'd like to just turn it back to the panelists for one last time around for closing remarks. So any closing remarks or thoughts? Jay, we'll start with you.
Yeah, well, I think it's kind of really exciting to hear that there are, so many different approaches, that the focus here was really about novel directions. And I think that's, definitely what we need at this point. I feel like in general, those low-hanging fruits, as I mentioned, of being able to just symptom manage largely for age-related diseases is taking a huge toll on our healthcare system at this point, and in all countries, not I'm speaking myself for stateside, but in all countries. And so having these more transformative solutions where you're trying to get root causes of aging or degeneracy, as opposed to trying to chase after each specific disease or indication, I think that can be a lot more transformative, and definitely is worth investing in the years to come.
Thanks so much, Jay. You've got one question in the chat function. So in the interest of time, I'll point your attention there so you can answer that. So Lorna, how about yourself? Closing thoughts.
Sure, yes. I mean, I'm also intrigued by the idea of synergies between these different approaches. I think that might be a really fruitful approach. My view on this is I think it's always going to be a much more sensible idea to be dealing with the root causes of multiple diseases than to be putting a sticking plaster on the effects of it. But of course, it's going to mean we're going to need to think of new ways of evaluating how these things work. So obviously, classically, trials, you'd be looking at outcome measures which are specific to your indication. And very few of those outcome measures will translate across different indications. Whereas these new technologies, I think we're going to need those. We're going to need new ways of evaluating whether or not these things are working that actually look in a lot more of a holistic way.
Great, thank you, Lorna. And Reenie, last to you.
Yeah, so thanks everyone. This has been a really interesting session, and I agree with my fellow panelists. I think that synergistic approaches, particularly to aging, are important. I think kind of targeting the root cause is important. I gave you kind of one insight, which is when to intervene in disease. I think the other that we've really learned from moving from extensive animal studies into people is how long it takes. It's not always clearly translatable from animal models into human subjects. And really, when you're going at the root causes, I think that you might have more durable effects and more meaningful effects, but it might take longer to get there.
Thanks so much, Reenie. So we're right at our time. Thanks to everyone for joining this session, and to our panel speakers. Jay, I see that you have joined us, so I'll turn the floor back over to you.
Great, thank you, David. So thank you for moderating such a great session, and thank you to Reenie, Lorna and Jay for joining us today and giving those great presentations. Great to see lots of questions in the chat and have a fully engaged discussion. So we do have the next session that's starting in the next couple of minutes. That is a panel discussion that is looking at the roadmap to commercialization. So examining the clinical and regulatory pathways for longevity therapeutics. So go back to platform and join that session that will start in the next 10 minutes. Thank you all again for joining us, and I'll see you again soon later. Bye.